Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of the show, bringing you another really awesome guest today involved in science and technology and biomedicine, creating a better tomorrow for many patients out there. On the show today, we have the honor of being joined by Dr. Alexander Shneider, who is a biotech entrepreneur and scientist with over 2 decades of experience spanning oncology, immunology, vaccines, translational medicine. He is the founder and chief executive officer of a company called CureLab Oncology, where he's leading the development of novel biologic agents designed to fight cancer through a very unique dual approach, both by stimulating anti-tumor immunity while also addressing the chronic inflammation that's also seen in cancer microenvironments and underlying disease progression. Their lead therapeutic candidate, which is known as Elenagen, has demonstrated really strong safety profile so far, as well as very encouraging signals in international phase 2 studies, and is being explored both as an adjunct to standard cancer treatments and for its broader potential in inflammation-driven and age-associated diseases. Over the course of his career, Dr. Shneider has advised biotech, pharma companies, as well as investment organizations across all areas of R&D, as well as strategy, licensing, tech transfer. He previously had founded the company working in the area of rational design of DNA vaccines, and ultimately this developed into a much broader concept. In academia, Dr. Shneider has also held professor-level appointments and led the genetic vaccine laboratory at Sechenov University over in Russia, where he helped establish a multi-center clinical research infrastructure focused on various diseases involving chronic inflammation. His research also explored evolutionary conserved RNA structure as a target for antiviral therapies, as well as next-gen vaccine design. So he's been quite prolific in research, as well as entrepreneurial work, which we'll be getting into, and he's extensively published as well. So a really exciting set of topics to get into today. Honored to have him with us to tell this story. Dr. Alexander Shneider, welcome to the show.
Thank you, Ira. You may add a couple of corrections. First of all, I'm not that young. I don't have over 2 decades of experience. Unfortunately, look how bold I am. I have over 3 decades of the experience, and I also have an appointment with University of Ariel in Israel, and the academic appointment which you mentioned belongs to my past. This is a current appointment, which I hope to turn into great biotech achievements usable for people all over the world.
Excellent. Well, I'm looking forward to discussing a lot of things with you today. Before, though, we get into CureLab and what you're doing there in terms of advanced gynecological ovarian cancers and a lot of the work you've done, take us back a little bit, Alex, because I love looking at our guests' early work. And, your dissertation, which had to do with these polycyclic aromatic hydrocarbons, it had a really interesting intersection between a lot of where we're going to be talking today in terms of cancer, inflammatory disease, overall immune dysfunction, and sort of the unique ecosystems that all this stuff plays around in. I'm just to hear a little bit more about your background and a little bit of, because you've been systems thinking from a very early stage in your career, and love to hear about how some of this started off.
boring, the most insignificant, the most nothing to be proud of part of my professional life was my dissertation. I came to one of the most prestigious universities in the world, Boston. 4 days later, my wife gave birth to our first son, Myron. So we stayed at home. I was waking up every morning, going to this prestigious lab, where and the head of the lab was with a Nobel Prize laureate in immunology, a legendary guy. So everybody was believing that I was coming every day to do something creative, and she's staying at home to do boring stuff. One day, and my life was I was waking up roughly 10 a.m., coming to the lab by 11 a.m., and then around 9 p.m., I was coming home, staying home for 2 hours, working with our son, and coming back to the lab to work till 2 in the morning, and then coming back to home. So one day I came home. She gives me this chicken and asks me, "How do you like it? Today I tried a new method and I don't know how would you like this recipe?" And I was so ashamed. I did nothing creative that day in that lab, in that university. What did I do? I took a standard scientific method. I took an object which was not analyzed by this method as of yet. On the crossover of this object and this method, I will come up with a paper, useful paper. People would reference this paper, and this is the majority of what molecular biologists, biochemists, and biologists, blah, 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 were doing that day. We did nothing creative. And she, staying at home, she gave herself a chance to be creative. And that moment, I start asking myself a question: how does science evolve? And I came up with a paper which was rejected 12 times by 12 journals. I was so emotionally damaged that I decided not to publish it. And when I decided to abandon that paper, I was sitting with a great economist, perhaps one of the most interesting economists of the 20th century in Los Angeles restaurant, who read that manuscript, and he wanted to discuss it with me because it was not only about biological science, it was about equally applicable to other sciences as well. And I told him, I don't want to discuss it. I decided to abandon this work. Too much pain, too much trauma, too much humiliation. And he told me, you know what? A Nobel Prize for asymmetrical information was given for a paper which was rejected 16 times. Mm-hmm. So let's make a deal. I will pay for our dinner for both of us, but you will drop this paper only after 17 rejections. And I laughed to myself, and I'm not only very poor, I'm also very greedy. So he said, okay, let's go somewhere in between. Not 12 times, not 17 times, but 14 or 15 rejections. And I sent it to, not. He also said, I sent it to my friend in Great Britain. I'm interested to learn his opinion. Okay. 4 o'clock in the morning in Boston, Monday, 4 o'clock, maybe 5 o'clock in the morning. phone rings. It was before I had always my cell phone next to me. But my stationary phone rings, 4 o'clock or 5 o'clock a.m. Every call is welcome at that time. Hello. And I hear a very prominent British accent. Good morning. My name is Jonathan Tyzik. I think Jonathan Tyzik, yeah. I'm an editor-in-chief of Trends in Biochemical Science tips, yeah, Trends in Biochemical Science. Do you do you realize that no review panel would ever let your paper through? I say, sir, do you call to every person 5 in the morning to tell him that his paper is rejection, or every other one? And you know what? Maybe some people would take the rejection news better after 9 a.m. Well, just think about it. Yeah. And he said, could you hear what I said? Did I say that your paper is rejected? I just told you that this paper will never be accepted by any panel of reviewers. I didn't tell you that this paper was rejected. I'm accepting it without sending it for a review. Wow. But you know what? Technically, you cannot submit this paper because electronic submission system has after a certain number of words, and you have exactly twice as many. I said, oh, really? I was so happy. I wanted this paper out of my life. I wanted to turn the page. I didn't want to have anything to do with it anymore. I said, okay, tell me what to take out. You ruined my weekend. I spent entire weekend thinking what to take out. And then I decided that it's easier for me, instead of thinking what to take out, to submit it through the back office. Really? Great. Hey, you know what, Alex? If you came not to the United States, but to Great Britain, at least you could learn better English. What can I do? You will have the best free secretary you could hope for. I will correct your as and these because you had a random number generator to put your as and these there. So that paper became the most read paper, according to Faculty 1000, for a long period of time. It held #1 place in hidden jewel category, and some journals who even rejected that paper for publication wrote editorials and took short interviews with me about that paper. Why I brought this? And today it has hundreds of references, not necessarily from only biological science. So I only brought up this story to tell you that the only strong part which my PhD thesis brought to my life is that I was perpetually ashamed for boring stuff I was spending my life on. And one of the reasons why I was compelled to start my own company is that I knew if I stay in academia, I would either need to become submissive, fighting for grants with, in statistics, there is a term diminishing to the mean, yeah? So everything comes to the middle point, where you cannot be behind, you cannot be outside, you cannot be. Academia does not allow you to look out of the box. You need to be among the first within the box. So either I had to stay in academia, or I had to create a little piece of a universe where it will be called it would be unpleasant, but at least I could create an environment where my teammates and I would try something outside of the current dominant academic dogmas. If we disagree with those dogmas, because I hate revolutionaries. We're not revolutionaries. If we agree with the dogmas, fine. But being within the dogma should be my freedom, not my obligation.
Well, in lieu of that, I still think it was a kind of a cool dissertation, but we'll agree it's not that. It's boring. Okay, it's boring.
Yes, it was. If you, what is your comparison group? If you compare what others did on the adjacent benches, yeah, it was a very solid comparing. But who do you compare yourself with? Do you compare yourself with what others have been doing? That I can honestly report to American taxpayers that I did appropriate their taxpayer dollars or on the research grants given a little bit above and a little bit beyond what was statistically expected. Yeah. But if you compare yourself with what you could create and what you didn't, what you could, people who you could save and you didn't, knowledge which you could generate and you didn't, if you compare yourself not to people on adjacent or desks, but with what you could do and you didn't, unfortunately, uh, unfortunately, uh, we live in a society which is now missing what I think was much more prominent at the turning of 19th and 20th century. For example, I remember when I was a young student, like maybe 19 or 20 years old, we were standing with few other not fratty looking guys in the university corridor. There were cool girls next to us. We tried to make a good impression on them. What was the way to have to make a good impression? Fake like you understand something in art, for example, impressionism, or to fake you understand something in quantum mechanics, which is an impressionism out of natural sciences. And we were talking about things we understood nothing about. And a young professor, I never saw him neither before nor after, was passing by. And he stopped and he turned around and he told us two sentences. I don't know what was his name, but those two sentences are with me every day, 3 decades, more than 3 decades after. And he didn't even pay attention to that few seconds episode of his life. He told us, guys, you are discussing the world in a very non-quantitative way. Why? To make your conversation more quantitative. This was the second less important part. But he also said, guys, Mother Nature is always much more complex and much more multifaceted than we model it. Yeah. Everything what you're discussing now has nothing to do with actual Mother Nature. It just reflects your comprehension. Why is it important? Why is it with me every day? Look what is the current dominant invasive dogma in drug development. In 1971, when Nixon announced a war on cancer, the dogma which came, and back then, it was a very progressive dogma, not anymore. Let's understand molecular mechanisms of how diseases develop, how diseases progress. When we understand molecular mechanism, we will go from a mechanism to a drug. So it became not but for drug development. It did work for infectious diseases, and for infectious diseases, pretty well. For cancer, very little. Very little. We spent so much money and we achieved so few good actual results that now it's time to ask ourselves, would it be more result if we didn't shackle ourselves within this dogma? Now, today is February 17th, 2026. February 17th, 2026, I'm talking to you and we look back at the scientists of previous eras and say, back then, we, by we I mean humanity, we didn't understand this, we didn't understand that, we misinterpreted this, we misinterpreted that. Back then, we didn't know yet this, and this. Okay. So we know how deficient our knowledge was yesterday. Hopefully, if our politicians did not start World War III, we have a chance to talk to 20 years from now, 30 years from now. Then we will look back at February 17th, 2026. And we will be as critical of our current knowledge tomorrow as we are critical of the knowledge of yesterday. Thus, an idea that you can win just only by rational design, mechanism-based drug development is a dead-end idea. Yes, we should do this research. Yes, we should incorporate those pieces here and there. But what we lost? We lost an ability to be humble when we talk about an unattainable, infinite complexity of Mother Nature. And am I proud of my leadership of CureLab? If yes, just for one thing, we do not, and we will not walk away from serendipitous observations. And if you honestly analyze history of greatest medical achievements, they were not from a mechanism to a drug. It was a Möbius strip. From a drug to a mechanism to a better drug. And if you say this, you are a pariah for academic establishment. And more than scientific knowledge, more than education, more than talent, you need to have guts to sustain for an unknownly long period of time an outsider lifestyle. Then if you win, like it happened to us. Now, when we have our clinical data, those who were the most disrespectful of you, they would claim that they were always on your side. Those who were not neutral, they would remain to be slightly positively neutral. But the elements which we are missing now more and more societally when we train new scientists is not a particular knowledge. American universities are the best in the world, providing students access to equipment, access to techniques, access to reagents. Graduate education in life sciences in America, wow, it's great. If you want, plus PubMed allows a student even from weaker school to have the same access to the knowledge, data. I think majority of professors would reply to an email with a particular question from a student from another university, because it would be very impolite not to reply to a question. The culture is good. What we're missing? We're missing human components, ability to be humble, readiness to sustain societal ostracizing, because woke catastrophe is not only on the cultural aspect of academic life. Woke catastrophe is also on the way how you do your research. What is and what is not to be funded, who is or who is not to be invited based on absolutely dubious parameters of measuring scientific productivity, and so on and so forth. So to me, CureLab, yes, we're fighting cancer. Yes, we're developing new era of medicine for anti-aging, anti-chronic inflammation. But to me, it's a very, do you remember Albert Einstein once said, there is nothing more practical than a good theory. So to me, it's a practical application of certain worldviews. One of them is reflected in the paper about 4 stages of scientific evolution and 4 types of scientists. Another paper, which is very actively used by some practicing companies. I know this because they're calling me and asking specific questions, but very rarely referenced. And if you would be interested, I will tell you why it cannot be referenced. The paper on overcoming mental inertia in biomedical sciences. To me, to our team, it's not a revolt. No, we're not revolutionary. It's a redemption from the box of contemporary scientific, applied, and basic scientific norms of the current society.
Well, that's a lot. But before we go into, it's again, you're laying the basis for, a lot of the last 50 years of research in this and related spaces. But let's really drill down and talk about where we're going, Alex, because the center of this complex and multifaceted story behind CureLab has to do with the gene called P62, a really interesting sort of, again, multifunctional protein that is involved in everything from autophagy, inflammatory and immune activation, nutrient sensing. I mean, it touches a lot of stuff. Um, introduce the audience to P62, also known as SQSTM1, what this gene normally does, and a little bit of sort of the first step. The initial hypothesis here was sort of tumor cells make a lot of this stuff. we're going to create a DNA vaccine and we're going to knock out those cells, but you found out something totally different. But talk a little bit about P62 and what that story is all about, and sort of the initial thinking behind a DNA vaccine that you found that does something totally different.
I was among those guys who started too early, too prematurely, direction called DNA vaccines. And the idea was like this. It wasn't. Is DNA or RNA vaccines is the same? There is very little difference. The idea was like this. Let's say there is a microbe and or a virus. There is a protein sitting on the surface of the microbe, or there is a protein which does not exist within this microbe, but if or a virus, but when a virus infects cell, this protein will be transiently produced within the cell. Let's take a piece of DNA, insert there a gene of this protein, and administer it to the body. So, when this gene is expressed, the body will produce enough, a lot of this protein. It will train immune system to recognize this protein and attack. Okay. Then, weeks, months, years, decades later, true infective agent comes. It doesn't know that we already trained immunity to recognize it based on this protein. So it will come and it will reignite, reactivate an immune response. And reactivating an immune response, it takes much less than to develop a regional primary immune response. Okay. And the problem was, it worked very well in mice and worked very poorly with bigger animals. And one NIH official told me in the corridor that my grants will never be supported because NIH is not funding a employment agency for space cadets. DNA or RNA vaccines will never materialize to anything other than mice work. And I asked myself, how can we make them work stronger? History of science and technology is teaching us that at each new level, technology repeats all the same steps it had at the previous levels. Let's look at the previous levels. At the previous levels, first we were giving different antigens, let's say proteins, and they worked very poorly. Then we start giving adjuvants. For example, alum. What is alum? It's a small colloidal size particles. Antigen is getting stuck on the surface, and that is very much more immunogenic. Now, when I don't give the protein, but I give a gene of the protein, I cannot give these small particles to go inside the same cell. And what can I do? What if I make these proteins to self-aggregate and become their own adjuvants and to form bigger particles as they are produced? And he said, okay, there is a disease called Huntington disease. There are certain tails if you fuse a gene of this tail to a fuse of a protein which you want to use the antigen, they will self-assemble. And yes, we developed a technique to make immune response much stronger, because each molecule of an antigen was immediately a molecule of its own adjuvant for self-assembling. And then I was invited with lectures, presentations, conferences, and I was using metaphors. I was saying, now these self-assembled particles go to lysosome, and then this, and this. But at night, when I was alone, I felt that I'm faking it. What does it mean, go? These particles don't have legs. They don't have free will to select a direction. And then I said, okay, there is a protein called P62. It grabs these particles and leads them to lysosomes. So this is how, working on something completely different, or working on vaccines, on anti-infective agents, I got P62 brought to my attention. And when I start analyzing it, I realized that this protein may become a great dreamlike antigen for therapeutic DNA agent against cancer cells. Why? Because an ideal target, ideal anti-cancer therapeutic vaccines would have two features. First, selectivity. Take a tumor, pour concentrated acid over a tumor, it will kill a tumor. Does it mean that concentrated acid is a good anti-cancer treatment? No. Why? No selectivity. It will equally kill any other tissue. So you need selectivity, number one. Okay. This protein, P62, is selectively overproduced in cancer cells, and cancer cells only. It is produced in all other cells, but not to the overwhelmingly excessive amount. And from immunological point of view, I believed at that time, and I even more believe today, that our immunity reacts not only to what kind of a target it is, but the quantity. Because when you have too many molecules of a particular protein, it starts forming other blocks, other partnerships, which have different immunological properties. So I said, okay, if cancer cells and cancer cells only have too much of P62, only in these cancer cells it would have different types of polyprotein complexes. They may be a different immunological beast. Okay, so selectivity is here. What is the nightmare number two? Nightmare of immunologists and nightmare of cancer therapy. That nightmare cost humanity hundreds of billions of dollars and millions of lives. And it is still, unfortunately, with us. A great British European mathematician Pascal said that people can do whatever, just avoid thinking. It's very simple to imagine that there is some magic lock as a target, and you just need to take a magic key specific for this target. and have a targeted therapy. It could be immunological target to develop immune response against this target. It could be a small molecule target, so like a ligand or derivative hits a particular cancer-associated receptor molecule. What is the tragedy of this approach? Imagine you have a picture of a patient with a tumor. There, you have a second picture, this this patient without a tumor.
Mm-hmm.
But you don't need you don't need to schedule your celebratory party because you have nothing to celebrate.
Mm-hmm.
Out of millions of more cells, there were few cells without this target.
Mm-hmm.
Out of millions of cells, there will be few cells who will omit producing this target, mutate out. So when you say no tumor anymore, it's no visible mass, but there are few cells which then will repopulate the tumor, and it will not contain this target, and your previous approach would be completely useless. So evading targeted attack, evading producing an antigen or you develop immune response against is the nightmare of immunologists and any targeted cancer therapy drug development. But with P62, cancer cells cannot stop overproducing it because they're addicted. Addicted not in social terms, but in biochemical terms. They can, for certain reasons, they're addicted to overproducing P62. If, in order to survive our attack, they have to stop overproducing P62, they would become so vulnerable to all other chemo, radiation therapies, and so on and so forth, that it would be a great desired result, desired outcome anyways. So he said, okay, now we take this circular DNA, it's called plasmid. You can analyze plasmid with an envelope, and you can put into an envelope a letter where you write any gene. In this case, plasmid backbone, plasmid molecular envelope. Circular DNA we're using as molecular envelope. We inserted a letter where we wrote a gene 62. It worked. Different product models, mice, rats. Then it was a very interesting story how it came to dogs. Dogs, much less so cats, because for some reason dogs are more likely to develop cancers. So we were with mice, rats, dogs, and now we have great results of in humans. But you know what is interesting? That although everything I told you is correct, it is more or less irrelevant. Because Mother Nature was much more multifaceted than I could model it to be, and we serendipitously run into discoveries that there are entirely different layers of the activities which I could not envision, I could not predict, and I was ridiculed for not walking away from studying them when we serendipitously run into them. I was told that I'm losing focus, which is a cursed accusation for a biotech CEO. But now I can tell you that the original idea is correct, but it's just a small piece of a much bigger puzzle.
And what you found out with Elenogen is not that you have a DNA vaccine against a tumor. You had something much broader that, yeah, I guess, you refer to it as a sort of a conditioning agent, a reprogramming the ecosystem, turning cold tumors hot, doing all sorts of other interesting things in the microenvironment. Ultimately, with this plasmid P62 agent, what do you, as you think about it, we'll get into the clinical data in a minute, but how do you refer to it? Systemic systems
Let me try to put it
immunomodulator,
in a
what is it?
simple way. Let me try to put it in a simple way, because I can answer the question what is it. I cannot answer the question how is it, because what is it is a statement which we can prove with experimental data. How is it? We can always remember that our knowledge is incomplete. So, what is it? When we inject our plasmid, by the way, it's important that the most beautiful wife in antiquity, her name was Elena. This is how Trojan War started, because the most beautiful woman in antiquity was Elena. The most beautiful woman today is still Elena. She's my wife, and we call this plasmid Elenagen, because this is my tribute to her. Without her, I would achieve nothing and I would develop nothing. So this plasmid with P62 is called Elenagen. So, and its derivative form for dogs and cats and horses called Elenagen. Very similar, a little different, right? It's like Toyota versus Honda, for example. So we take Elenagen, we inject it, and it rejuvenates stem cells in the body. For example, when you're young and sporty in your bones, stem cells develop osteoblasts, bone cells. When you become a grandpa and your grandchildren bring their children for your birthday party, unfortunately, your stem cells mostly develop not osteoblasts, but adipocytes, fat cells. That's why your bones are so fragile when you are, okay? When you're young and healthy, your stem cells secrete anti-inflammatory cytokines, anti-inflammatory signals. It's called secretome. When you hear it means I don't know what the hell it is, but it's a collection of everything what is there, okay? If secretion, it's called secretome. It means everything they secrete, okay? So, unfortunately, when you become a grandpa or grandma, or you're becoming ill, instead of secreting anti-inflammatory, they start secreting pro-inflammatory secretome. And it travels all over the body and causes many ugly things. You can call it aging, but in each particular manifestation in skin, it is psoriasis, many cancers, bone diseases, Alzheimer, everything. So when we give Elenagen, it reprograms stem cells to act like they're younger, to stop secreting pro-inflammatory and and again start secreting anti-inflammatory secretome. If you will be interested, I'll tell you later, how did we find that Elenagen does it indirectly and how it self-amplifies within our body, but as an effect, not as a molecule. But at the first simple level, we give Elenagen, we reprogram, somewhat rejuvenate stem cells, we reduce chronic inflammation in the body systemically. in the tumor. First of all, chronic inflammation in the tumor makes a tumor. Okay, first of all, please allow me to take a little bit of step back. Tumor is not just a bag with balls called cancer cells. Tumor is a very tightly organized, a very delicate architectural structure with corridors and streets, call them extracellular matrix, and some streets are good for certain cells to penetrate, some streets are good for certain cells like metastatic cells to exit. So the entire structure, intratumoral structure, is very good for metastatic cells to exit and very bad for immune cells to penetrate. It's called cold tumors. It's a slangish word. Their temperature is the same as the temperature of the rest of your body. Cold means no lymphocyte penetration there. Now, humanity spends hundreds of billions of dollars on all sorts of immune therapies, cell therapies, CAR-T, blah, blah, blah. The problem is you can produce an unlimited amount of lymphocytes which are very effective to kill cancer cells. There is one little problem. In order for an immune cells to kill cancer cells, they need to meet at some point. In order for them to meet, an immune cells need to penetrate a tumor. If an immune cell cannot penetrate a tumor, this entire therapy is the most expensive, useless thing. We change intratumoral structure. So now these immune cells can penetrate. And now we do not come instead, we come in addition to those great minds who made great achievement, but they were lacking this one element. And we will be used together with all other therapies, chemotherapy, radiation therapy, immune therapy, because all of them are immune therapies for us. Mm-hmm. Mm-hmm. There is no chemotherapy. There is chemo-induced immune therapy. There is no radiation therapy. There is a radiation-induced immune therapy. So first, we reduce chronic inflammation with open tumors. So now immune cells can penetrate. What else? Imagine, actually, it was described in epic literature centuries, if not millennia, before us, when warriors come to a palace and they hear such a beautiful singing that they fall asleep, and then much weaker soldiers within the palace come and cut their throat. Okay, the same story happens in a tumor. Immune cells come, but in perpetually inflamed tumor microenvironment, put them asleep, induces their suicide, or induces their inactivation. It's called intratumoral immune suppression. There are multiple ways: ligand, extracellular adenosine, checkpoints, many things. All of them have a common denominator: chronic inflammation. Mm-hmm. We eliminate chronic inflammation, so not only we open the gates for immune cells to penetrate, not only we change the carpet on the intratumoral floor that metastatic cells have harder time to exit. Also, when these warriors, immune cells, come in, they remain active. And this is much more important than my original hypothesis, because my original hypothesis was just about one great, beautiful, efficient, but just one tumor-associated antigen, P62. Now, it turns out that the same P62, other than being an antigen, it activates, reactivates, reignites effective immune response against all other countless tumor-associated antigens, which we don't know about. We don't have to know about them, but they work with us. Okay? Okay, terrific. Also, in our last paper, maybe it was the most important paper of my life so far, this achievement with ovarian cancer, deadliest form of ovarian cancer. We combined Elenagen with gemcitabine.
Mm-hmm.
With gemcitabine and many other drugs, they're self-limiting. Why do we give them? Because we want them to do a particular thing, anti-cancer activity. And we measured them for this activity. Unfortunately, other than what we keep in mind, there are things which we didn't take into account. And what we didn't take into account, then they activate chronic inflammation. And chronic inflammation deactivates these drugs. So they're self-limiting. They come, they activate chronic inflammation deactivates the drugs. So even if they were effective for a limited duration of time. What Elenagen does, it blocks chronic inflammation. So gemcitabine or other drugs, they will remain to be able to do what we want them to do, which is what? Manifest anti-cancer activity, but they will not be able to activate self-limiting negative feedback loop. Listen, this is already what we know and what we understand today. But tomorrow, I may look back and say, yesterday I didn't understand that was insignificant. Correct, but insignificant. This is, I think, the most significant. Tomorrow I may look back and say, yes, that was correct and insignificant. Then the second level also was correct, but now I also know it's insignificant. Now I know something else is the bigger part of how it works. And this is perhaps as much of a differentiator between our company and what is socially accepted today in the industry, as much as our highly encouraging, not to use a stronger word, clinical results.
Well, let's talk, Alex, a little bit about those because, again, you did the recent coming off the phase 2 study, and this is in the worst of the worst, as you were mentioning, platinum-resistant ovarian cancer, combined with gemcitabine, some pretty impressive results in terms of nearly doubling progression-free survival here. So it took us into a little bit of what you learned here, because this is a recent study. We'll put a link to it. But again, showing the, in the initial context of what you've been working on, that there is a certain human translation here.
First of all, let's define the target group. With ovarian cancer, there is a marker. It's called CA125. Women who have this level of CA125 above 35, they're the worst prognosis women, the worst prognosis patient. Lower let's say 8 or 19, it doesn't matter. They're much better prognosis, much more treatable, live longer. So we only talk for the next couple of minutes about women with the worst prognosis. Because imagine a fictionary hero who comes and says, I won against 98% of animals I fought against. And I fought against elephants and other animals. Then you learn that 98% were mice and only 2 elephants. And the truth is, yes, they're all animals. One of them, they're all mammals. And he honestly won against 98% of animals. He didn't win against only 2 animals, elephants. But in the group containing animals, he did win against 98% of mammals. So, to avoid this self-indulgence, we said, we don't take all patients, we take only high CA125 patients. And of course, as always, when you raise the bar, you will, they're called patients, they're called doctors, who would be thankful. And there are those who will now hate you for that, because now when the bar is raised for yourself, it is also raised for them. They didn't ask you to ruin their careers. They take you as a passive aggressive. Okay, so we're only taking high CA 125 patients. In an oversimplified way, without going into nuances. At the beginning, ovarian cancer patients are treated with platinum drugs. Alone or in combination with paclitaxel, let's say typically 6 courses of platinum, and many women, maybe even majority of them, not all, would be very responsive. For some of them, for a prolonged period of time. Unfortunately, there are 2 problems. First, there is a certain segment of women who will not be responsive, and they also want to stay alive. Their tumors are the most aggressive. They're so aggressive that platinum doesn't work. And there is also unfortunate truth that even those women who were responsive, sooner or later, cancer will come back, and it would be impossible to save them with just platinum drugs again. It's called platinum-resistant ovarian cancer. If you're interested, I will tell you a very interesting true story how platinum drugs were discovered because it had nothing to do with how it is presented in medical textbooks. But anyways, we take these women, platinum-resistant ovarian cancer with high CA125, and we treat them with standard of care plus or minus Elenogen, just either standard of care alone or with Elenogen. Unfortunately, due to military political situation, in July 2022, we could not bring new Elenogen to the clinical sites. So in July of 2022, all women stopped receiving Elenogen. By this time, some of them received it for a very short period of time, less than 1 month. Some of them were receiving Elenogen already for a very long period of time, like 30 months, and anywhere in between, because it was just synchronous termination, regardless of the pre-existing history. So, oncologists were continuing treating these women, those who didn't receive Elenogen and those who did receive Elenogen, with absolutely the same treatment, and observed for how long will they live. And it turned out that women receiving standard of care lived in average for 13 months. This is what other people observe. Somebody says 12, somebody says 14. It's a very small difference. So what we had in our standard group was very standard for the world. But if women received Elenogen, it was not 13 months, but 25 months. Wow. But now, because of the war, we had a serendipitous experiment, which otherwise I would never do. Can we correlate if the duration for how long Elenogen was given influences for how long a woman would live after it was stopped? Once again, we're not Gestapo or SS. Of course, we would never do it intentionally, such experiment. It would be inhumane. But we had this serendipitous data, and this is also part of the paper. And we see beautiful correlation that for first 12 months of treatment, the longer a woman receives Elenogen, for longer she will live after it is terminated. Then between somewhere 12 and 18 to 24 months, the effect of longer treatment dissipates. Yes, it is still positive, but a marginal benefit of one more months of treatment is smaller, and smaller. So now we know that in the future, first of all, we will treat women with Elenogen for 18 to 24 months. Second, we know that we will see an effect much stronger than what we reported now. The vast majority of the women we report now received it for much less than 18 or 24 months. Okay? So we now know that yes, it works. Yes, it prolongs life. We now know that in the future, we will administer Elenogen for somewhere between 18 or 24 months. And we now know that it is imperative to repeat and extend this experiment in the United States of America and expect much greater life-prolonging effect. And what is even more important, quality of life. In the future testing, we will officially include monitoring of quality of life because although it was not part of a protocol, several women self-reported greatly improved quality of life, which is not surprising. I mean, of course, there is mental effect that you live longer, you feel better. But also, there is a purely biochemical reason, many side effects related to different forms of chronic inflammation. You reduce chronic inflammation, you both reduce toxicity caused by standards of treatment, but you also reduce comorbid diseases and comorbid manifestations which reduce quality of life. So this is the journey from purely laboratory thinking to actually bedside. And perhaps I was lucky to have this journey because mostly people see only one episode of the saga. And I was lucky to observe and walk through and continue walking through the entire path.
And Alex, take us into 2026 and where things are going. So, obviously, there's going to be continuation of the ovarian cancer work on these larger populations. You're going to extend it to different countries. You have the animal
The
health
country I
division.
love, called the United States of America.
Yeah.
And why should I limit myself by reducing pain and suffering of women across the pond if tears of women here are as salty as tears over there? The goal number one is to bring it here. We just received, by the way, a score called, score 20. It's an excellent score from NCI for grant application for this research. More likely than not, with such an excellent score, we hope to receive for the first time help from Uncle Sam to do this, and that would be done on American soil. So this is because, listen, I feel myself like a passive murderer. I'm getting dozens of calls every week from people urgently needing help, and they say, how can we get into the clinical study? And there is no clinical study, and we have no right to procrastinate, because for this man, his daughter will not live long enough if we procrastinate. It's not a universally happy experience, but it's a multidimensional picture, multidimensional journey. Journey dimension number one: just bring what you've done over there, bring it here, repeat, extend, and help Big Pharma, help Uncle Sam to bring it to women in need. I mean, this is a no-brainer. Number two, try other combinations. Why did we combine with gemcitabine? Because this combination has a divine origin. Alex Shneider had to say something. Mm-hmm. But what does Alex Shneider know? Alex Shneider knows nothing. Maybe tomorrow, a combination with Clitoxel or Daxil will work better than with gemcitabine. Then I will lie to you that, I envisioned that it would work better than gemcitabine for retrospectively invented reasons. But this is how mechanism-based drug design works. But so dimension number two is try other combinations. Dimension number three, try other cancers where gemcitabine is a part of their standard of care. Try to to bring Elenogen there. Dimension number four, take other cancers. We talk only about solid tumors so far. We're not talking of sarcomas or only carcinomas. We're not talking of sarcomas. We're not talking blood cancer. Focus on what you claim you know something about. Take other cancers where chronic inflammation is an important part of pathogenesis. Try there. Another dimension, take uh, diseases of chronic inflammation, which you already have either animal data you published or some case reports in humans, and try to apply there.
Mhm.
Of course, we will need to cut out this part because otherwise I would be accused of being unable to keep focus, and this is a curse. Uh,
No, not really. I mean, look, I think
yeah,
we've had enough guests
joke,
on the
man.
show that
Don't be too serious. You know, the biggest mistakes humanity made with serious faces. So if you look, all worst political decisions, they're always made by politicians with no smile. You need to go step by step. Like, for example, we win first battle with ovarian cancer, then we reappropriate money for other. Now, this is one universe. There is another universe called cats, dogs, cats, much less so horses. There, for many people, they are four-legged family members. We saved 10 out of 11 dogs with breast cancer. We blocked metastatic process. By the way, not the primary tumor, but it's more likely can be resectable with oral melanoma with dogs. We published a paper with videos. You can see an aging dog is limping, cannot put its weight on one of the legs, and after 8 injections, it's running like crazy, feeling young. So you don't need to be a veterinarian to see that we reduce chronic pain associated with limping and decline of activity. Soon we will have another paper uh, on other canine disease of chronic inflammation, which we're about to submit. So there is a human universe, and for that universe, CureLab Oncology is responsible. And there is another company called CureLab Veterinary. It's responsible for veterinary universe because we as a society become more compassionate, more inclusive. And now, for the first time in human history, we can show compassion not only to humans but to animals as well.
Yeah, that's Biotech 101, but the way you're thinking about it obviously is extremely important, and understanding both the companion animal market, which is a big one, but obviously going step by step, starting with what you have and growing it from there. Obviously, you can't do every indication at once, but I do appreciate the underlying nature here of immunology and inflammation and immunosenescence and everything that comes in that package that underlies a lot of these chronic diseases. So it's an exciting story, and the fact that it started off in one direction and went in a totally opposite one. Well, not really an opposite one, but as you said, it's doing something. We don't know why, but um, you know, you'll learn a lot lot about that on the as you continue
Well,
to move forward in
we
2026.
are learning the mechanisms. We are learning through this lens
Right.
where we are learning about certain biological phenomena behind it and how it works, opens the curtain, which now allows us to look uh, behind the scene and see how it works and then we hope it would become part of the just basic science biological knowledge uh, because I believe there is only one difference between true applied scientists and true basic science scientists. And the difference is internal motivation. For example, one physicist was thinking, I want to learn how atom nucleus is designed because I want to understand the mother nature. Another guy said, Hitler killed so many people I loved. I want to drop an atomic bomb on him. Internal motivation is different. Then they do absolutely the same calculations, absolutely the same experiments. One is applied scientist, one is basic sciences. One guy says, I want to study biology just to understand how it works. Another one says, she died. I'm afraid that my friend will outlive his woman and suffer as much as I do. I want to develop a drug. Then these two guys do absolutely the same experiments, absolutely the same methods, but the internal motivation, one is purely nosological, to learn how the world works. Another says, without understanding this element, I would not be able to develop this product, whatever the product is, a vaccine to prevent a disease or an atomic bomb to kill an enemy, doesn't matter. The difference between an applied and basic scientist is an internal motivation. The rest is the same. So we hope we see a big multidimensional Möbius strip when humbled drug development, not from a mechanism to a drug, but from a drug to a mechanism to a better drug, will also generate knowledge for the sake of knowledge.
Alex, what else is coming up for 2026 that we should know about while we have
My
you
wife
today?
and I will dance on a tango festival. So this comes for 2026. I'm sure this was your question, was it not?
The company.
For the company, let me tell you what is not coming. I'm not giving any forward-looking statement, which then would be incomplete, and then some guy who will buy one share will sue my behind for violating fiduciary duties. I am too old and too experienced not to fall into this unintended trap. I give no forward-looking statements, but I can tell you that we will put our entire sweat and tears into implementing the program I just mentioned a few minutes ago. We're devoted and motivated to this. We will be, during 2026, we may be opened for pre-IPO investments. If you know Cassandra, send her to my way, because she would know whether it would or would not make sense for us to make an IPO, because it would depend on such many factors, including macro political and stuff like this. And for some reason, neither Trump nor Xi Jinping are calling me to ask for permission to do something. By the way, I would not allow many things, but it's unpredictable. We will be current with what is going on. And if reality changes, keeping the same goal, the same focus on the R&D, we will always twist our tactics, keeping our strategy. The strategy is perpetual. The tactics, every morning I wake up and I'm ready to make a twist if I open my computer and see that reality changed. So you want to make God laughing? Tell him about your plans.
Well, I wish you the best for 2026, Alex. I look forward to seeing the progression of the company and the programs. Again, for everybody out there that's going to be listening to this episode across the podcast networks or watching on the YouTube channel, you've been spending time with Dr. Alexander Shneider, founder, chief executive officer, CureLab Oncology. Alex, I want to thank you for taking the time out today to come talk to us, update us on your programs. Thank you for everything you do for patients. And as we like to say here on our show, thanks for helping to create a better tomorrow for people via the development of these products. Awesome story, and I look forward to continue to follow it.
Thank you for being such a great host, and thank you for an opportunity to meet with your audience. I'm honored and humbled to talk to you guys. I don't see your faces, but whatever we do, if we succeed, we do for you and your families.
Wonderful.